Hi dear students, a warm good morning to all of you and welcome to one more session of microbiology. Today we are going to talk about the bacterial ultrastructure. As we all know, bacteria does have a central nucleus followed by a cytoplasm, and surrounding the cytoplasm it does have a plasma membrane. In the previous sessions, live sessions which we had carried out, we had explained about the nucleus and various other organelles within it. And I told you about the plasma membrane which has been surrounding the what? The cytoplasm. If you go to consider the plasma membrane, it does contain a phospholipid bilayer. And unlike the other plasma membranes, which we find in plants as well as in animals, they do lack, the bacterial plasma membrane does lack cholesterol, and instead of that, it has hopanoids. But you can also find integral proteins as well as peripheral proteins attached to the plasma membrane also. So, the plasma membrane plays an important role in the case of bacteria for the transport of various materials across the barrier into the cell. As we had already discussed the plasma membrane in details in other sessions, I am not going into the details. And outer to the plasma membrane, the bacteria does have a cell wall. The cell wall is made up of peptidoglycan. And we all know that based upon the cell wall structure, we have divided bacteria into gram-positive bacteria as well as gram-negative bacteria. Both in gram-positive bacteria and gram-negative bacteria, the cell wall do contain peptidoglycan layer, but the thickness of the peptidoglycan layer is being found to be different in the case of gram-positive and gram-negative bacteria. While it is thick in gram-positive bacteria, as we can see the brown color here, it has been found to be thick in gram-positive bacteria, and it has been found to be thin in the case of gram-negative bacteria. And in the case of gram-negative bacteria, you can see that in addition to the peptidoglycan cell wall layer, they also do have a outer membrane. And it is on the presence of this outer membrane that the bacteria gets classified into gram-positive and gram-negative. Today in this session, I would be mainly explaining the difference in the the cell wall structure of gram-positive and gram-negative bacteria. Both of them do have a common peptidoglycan layer, and when an outer membrane is being found additional to the peptidoglycan layer in some bacteria, they are considered to be as gram-negative bacteria. And in between the plasma membrane and the cell wall, there do exist a space called the periplasmic space. In the case of gram-negative bacteria, this periplasmic space also includes the space between the peptidoglycan layer and the outer membrane. So, if you go to look into the peptidoglycan structure in the gram-positive and in the gram-negative bacteria, they do contain, it is a network or a mesh-like structure is being formed using the peptidoglycan cyclus, and it is referred to as a peptidoglycan cyclus. Now, what is the term peptidoglycan itself? From when you split it itself, you can see that it does contain peptide molecules as well as a glycan molecules. That is, protein-based molecules as well as sugar-derived molecules are also being made up of, are being found in the peptidoglycan structure. The two sugar derivatives mainly found in the peptidoglycan structure mainly includes N-acetylglucosamine and N-acetylmuramic acid. You can just simply call it as NAM and NAG. NAM refers to N-acetylmuramic acid and NAG refers to N-acetylglucosamine for your convenience. These sugar derivatives are alternatively placed in chains, and this chain is being arranged in a helical fashion to form a peptidoglycan structure. And attached to this NAM or N-acetylmuramic acid, there would be certain amino acids attached. And these amino acids will be protruding out from the helical structure. And they include D-glutamic acid, D-alanine, mesodiaminopymalic acid. And because D amino acids have been present, most of the peptidases which could normally act on the L amino acids will not act on these peptidoglycan cell wall of the bacteria because they do contain D amino acids. You can also find that sometimes L-alanine amino acid is also being found. So this is how the cell wall is being made up of. That is, alternative sugar derivatives of N-acetyl glucosamine and N-acetylmuramic acid. They have been arranged in chains and attached to the NAM molecule. Yeah, on the N-acetylmuramic acid, there are amino acid chains which are being attached. Like I had told you about, which are the amino acids which we found. We have either D-glutamic acid, D-alanine, mesodiaminopymalic acid, like that. Different amino acids are being attached. A chain of 4 amino acids will be attached onto the NAM. And like that, what happens? Each peptidoglycan chain is being interlinked with different peptidoglycan chains by peptide bonds. And sometimes they might be pentaglycine interbridges. Now in the case of gram-positive bacteria, 5 glycine moieties will be combined together and they will form a peptide interbridge over here. So if you go more into the details, you can see that every NAM and NAG, see, there is alternative NAM and NAG. In every chain, they are being arranged in a helical fashion, and attached to them we have the amino acids side chains which are being attached, and they protrude out from the what? From the main chain of NAM and NAG, and either they will be at 90-degree angle or sometimes at 120-degree angle protruding out from the NAM and the NAG. Now, each of these helical molecule, it mainly refers to this particular chain. It is not a straight chain here, but in fact, it is a helical chain which is being arranged like that. And attached to that, we have the amino acid chains. If you want to have a more closer look, you can have NAM and NAG, which having 4 amino acids attached to it. You have L-alanine, D-glutamic acid, mesodiaminopymalic acid, as well as D-alanine. So this is the peptidoglycan chain, the amino acid chain which has been attached to the peptidoglycan in gram-negative bacteria E. coli. Now, if you go to consider the gram-positive bacteria and the gram-negative bacterial cell wall, the peptidoglycan structure has been found to be slightly changed in base of the amino acids which have been attached to it. In the case of gram-negative bacteria, it is L-alanine, D-glutamic acid, diaminopymalic acid, and D-alanine. And you can see that in the closely associated peptidoglycan chains are being interlinked together by some bonds. And this is mainly being attached by the forming of a peptide bond between the 4th amino acid, D-alanine, especially the COOH of D-alanine, is mainly linked to the penultimate or the 3rd amino acid of the next chain. Here it is DAP or the diaminopymalic acid. So the NH2 group of DAP is being attached to the COOH group of alanine by a peptide bond to form the peptide interbridge. Now what is this peptide interbridge? You can have a look here. It is similar to this blue color which is being depicted over here. And in the case of gram-positive bacteria, it has been found that these amino acids are being replaced, and you can find that instead of DAP, we have L-lysine. And instead of the peptide bond, you have a pentaglycine chain which has been linking between these. So that's how the gram-positive and the gram-negative bacterial cell wall, especially the peptidoglycan structure, will be found to be different. And you can see that various other diamino acids are also being found in the peptidoglycan. Like you can have lysine, diaminopymalic acid, as well as ornithine is also being present. So we will continue. In a gram-positive bacteria, you can see that the cell wall mainly consists of a thicker peptidoglycan layer, which has been shown by here. So then you have multiple layers of NAM and NAG, which have been arranged together, and it has been found to be very thick. And in between, they do have the peptide interbridges, which are as being depicted like this. And inner to the peptidoglycan layer, we have the periplasmic space, and inner to that, we have the plasma membrane, which already contains the integral proteins, peripheral proteins, as well as the phosphobilipid layer. And attached to this peptidoglycan layer, you can also find sometimes some molecules called the teichoic acids. And these teichoic acids sometimes will be linked towards the lipid layer down, that is in the plasma membrane, and when it is being linked to the lipid bilayer down, the teichoic acid is referred to as a lipoteichoic acid. Now, what is teichoic acid? Teichoic acid is a polymer of glycerol or ribitol joined by the phosphate groups. See, this is glycerol, the one which we use in tears and all that, glycerol, and this is ribitol. It is a 5 pentacarbon with OH groups. So a polymer of glycerol, ribitol, as well as when it is along with some phosphate groups, will contribute towards the formation of teichoic acids. And amino acids such as D-alanine or sugars such as glucose are attached to the glycerol and the ribitol groups in teichoic acids. Now, what is the function of these teichoic acids? Teichoic acids, as I mentioned earlier, they are either attached to the peptidoglycan layer or sometimes to the below lying lipid layer also. And they give negative charge to the cell, and hence the gram-positive bacterial cell wall will get a negative charge by the presence of the lipoteichoic acid or the teichoic acids. And the exact function of these teichoic acids are still unclear, but they play an important role in the maintenance of the structure of the bacterial cell wall. And do note that the teichoic acids are not being present in a gram-negative bacteria, but they are only being present in a gram-positive bacteria. Now let's move on to the periplasmic space in the gram-positive bacteria. You can see that the periplasmic space of the gram-positive bacteria lies between the plasma membrane and the cell wall, and it is smaller than that of the gram-negative bacteria, because we know in the gram-negative bacteria they also do have an outer membrane, and there is also a space between the outer membrane and the and also the peptidoglycan layer. And the periplasmic space of gram-positive bacteria do contain relatively few proteins, and they are mainly being involved because the peptidoglycan sac has been found to be porous, and many proteins secreted by the cell usually pass through it. They are usually considered to be as exoenzymes, and they often they play an important role in to degrade the polymeric nutrients that would be otherwise too large and to be transported through across a plasma membrane. So if we normally look onto the plasma membrane proteins, the plasma membrane proteins are the ones which play an important role in the transport of the nutrients across a plasma membrane. So the proteins in the periplasmic space, in the case of gram-positive bacteria, they would be probably the ones which serve as exoenzymes, and that is which will be excreted outside of the cell and which will help in the degradation of large polymeric nutrients so that they can be easily passed across the plasma membrane. And within the peptidoglycan layer also, we do have some proteins attached to it, and some of them are being found to be non-covalently linked, and some of them will be considered to be as covalently attached. And you can also find membrane-bound enzymes called the sortases, which will catalyze the formation of covalent bonds that join these proteins to the peptidoglycan. And
if we go to consider the other proteins like the covalently linked ones, you can see that some of them also play an important role in the virulence of the pathogen. What do you mean by the virulence? The virulence means the ability of the pathogen to cause a particular disease. Now, for example, M protein of the pathogenic streptococci aids in the adhesion to the host tissues and interferes with the host defenses. So that's about the gram-positive bacteria and its cell wall. Now let's move on to the gram-negative bacteria. As I mentioned earlier, the gram-negative bacteria does contain an additional outer membrane. That is, the plasma membrane is there, a thin layer of peptidoglycan which has the same composition as that of the NAM and NAG chain and all that as we saw in the gram-positive bacteria, but it does not have the teichoic acids. And outer to this peptidoglycan layer, we do have another structure called the outer membrane in the case of the gram-negative bacteria. Now, the outer membrane, it mainly has different parts and it is linked to the cell using a protein called the bronze lipoprotein as well as by certain sites called the adhesion sites. The bronze lipoprotein is the most abundant protein in the outer membrane and it is covalently joined to the underlying peptidoglycan layer and it is also embedded in the outer membrane by its hydrophobic end. And in addition to the bronze lipoprotein, we have also the adhesion sites which will join the outer membrane and the plasma membrane, which is mainly they may be by direct contact or truly by true membrane lesions. So just look here. If you want to attach this outer membrane onto the what underlying outer membrane peptidoglycan layer, either you can have some proteins called the bronze lipoprotein. Just notice this. See, the outer membrane is attached to the peptidoglycan by the bronze lipoprotein, or at some regions. You see, this is also bronze lipoprotein, and all that. So, in addition to this, at some regions, the peptidoglycan layer and the outer membrane region will be joined together by directly linking, and those regions are called the adhesion sites or the sticky sites, if you say in your own language. Okay, but the term adhesion sites is the most one appropriate. So, if you go to consider the outer membrane, it is mainly consisted of lipopolysaccharides. That is, lipids as well as polysaccharides are the ones which have been found to be more predominant in the case of the outer membrane. And it is a large complex molecule which contains both lipids and carbohydrate. And it mainly consists of three parts, the lipid A, core polysaccharide as well as the O side chain. Now, lipid A is a region which contains two glucosamine sugar derivatives, each with three fatty acids and a phosphate or a pyrophosphate attached. Lipid, as you just remember, in the case of lipid is a fatty derivative. Okay, in that you will have either glucosamine sugar derivatives and three fatty acids. When a fatty acid, and also you will also have a phosphate or a pyrophosphate attached to it. And the fatty acids of the lipid A are embedded in the outer membrane while the remainder of the lipopolysaccharide is projecting outside from the surface. The core polysaccharide is also joined to lipid A and the O side chain or the O antigen is a polysaccharide which is extending outside from the core, and you can see that it has peculiar sugars attached to it and varies composition is being found between different bacterial strains. Now let me just show you the structure so that you will see. So if you go to consider the outer membrane, as I told you, it mainly consists of three parts. The main composition is lipopolysaccharide, and from the lipopolysaccharide you do have a core antigen, core polysaccharide, lipid A and the O specific antigen. Now in this figure you can see the O specific antigen is a one which has been found to be protruding out. And you also have the lipid A. Now, lipid A here is, you can see the lipids. These are the lipids A. And attached to the lipid A, you do have the next one called the core polysaccharide here. And from that, the thread-like structure called the O-specific side chain will extend. The lipids are these ones which are having the hydrophobic head and the hydrophilic tails. Okay, these are the lipids. Attached to that, we have the core polysaccharide, and extending from that, we have the O-specific antigens, as I mentioned, which contains different sugars. Now, what is the contribution or what is the function of these lipopolysaccharides? They mainly contribute towards a negative charge on the bacterial surface, and they help to stabilize the outer membrane structure, because the lipid A is a major constituent of the exterior leaflet of the outer membrane, and it contributes to the bacterial attachment to the surfaces and biofilm formation, and it creates a permeability barrier. That is, only needed ones will be, only substances of good use of the bacteria will be permitted, the others will be inhibited. And the geometry of the lipopolysaccharides and interactions between the neighboring lipopolysaccharide molecules are thought to restrict the entry of bile salts, antibiotics, and other toxic substances that might kill or injure the bacteria. And you can also see that lipopolysaccharides, they also do have a role in protecting the pathogenic gram-negative bacteria from host defenses. And other than the lipopolysaccharide, you can see that the O side chain of this lipopolysaccharide is also called the O antigen, because it elicits an immune response by an infected host involving the production of antibodies that would bind to the strain-specific form of the LPS. Now, what do you mean by O antigen? O antigen is a component which has been found in the outer membrane of the bacteria. And if that happens to enter into the body of a host, what will happen? It will trigger the immune system. The immune system is the one which will protect us from infection. So if any component, or like if this O antigen of the bacteria happens to enter into our body will start producing antibodies against it. And the lipid A portion of lipopolysaccharide is found to be toxic, and it can serve as an endotoxin, and it can cause the symptoms which will arise due to the presence of the gram-negative bacterial infection. And if lipopolysaccharide or lipid A enters into the bloodstream, a form of septic shock will develop, for which there is no direct treatment. So such is the importance of this lipopolysaccharides. And attached to the outer membrane, we also do have another protein called the porin, and they mainly aid in the passage of the small molecules such as glucose and other monosaccharides. And sometimes you can see that most of the porin molecules might cluster together or group together to form a trimer in the outer membrane. And this porin will span from the outer membrane, and it has been considered to be more or less tube shaped. It will have a passage of molecules smaller than 600 to 7,000 daltons, and larger molecules such as vitamin B12 can also pass across the outer membrane. And you can see such molecules will not pass through the porins, but instead there would be other specific carriers which will transport across the outer membrane. Now let me just show you the porins present in the outer membrane. These are the porins which will aid in the transport of particles across from the outer membrane towards the inner side. So you have the outer membrane, which is made up of lipopolysaccharides consisting of lipid A, core polysaccharide, as well as the O side specific antigens, and then we have the porins attached to it, which will help in the transport of particles. And the outer membrane is attached to the underlying peptidoglycan layer through the bronze lipoproteins or through certain sites called the adhesion sites. Now I would like to talk about the periplasmic space in the gram-negative bacteria. As you know that the periplasmic space is the space between the peptidoglycan layer and the outer membrane and the peptidoglycan layer and the plasma membrane. And some periplasmic proteins in gram-negative bacteria do play an important role in the nutrient acquisition. Now, for example, hydrolytic enzymes like ATP, GTP, and also in the transport of nutrients. And there are various transport nutrients also being present. And sometimes they might be involved in energy conservation, that is to store enzymes, energy, or sometimes in the peptidoglycan synthesis or in the modification of toxic compounds that could harm the cell. So various proteins have been present in the gram-negative bacteria and they do play a different role. And if you go to compare the cell wall of the gram-positive bacteria and the gram-negative bacteria, the gram-positive bacteria has a 20 to 80 nanometer peptidoglycan layer, which has been found to be thicker and it will not contain an outer membrane. But in gram-negative bacteria, it is 2 to 7 nanometer with a outer membrane which has been present. And the thicker peptidoglycan layer, the walls of the gram-positive bacterial cells are more resistant to osmotic pressure than those of the gram-negative bacteria, but gram-negative bacteria has been found to be considered to be less resistant to osmotic stress. And the gram-positive bacteria, they do have peptide interbridges. Uh, Here, they lack peptide interbridges, but instead they have direct cross-linking between the uh, amino acids. You do remember that, I guess, the DAP and the DAP and the this one. So you can have a look over here. This is just one sec. This is the case of here. The DAP and the D-alanine, they have the direct bridges, the direct linking in the case of the what? Gram-negative bacteria. But here, in the case of gram-positive bacteria, they do have the peptide interbridges. So that was the point which I was explaining to you right now. And other than that, you can see that diaminopymalic acid has been present in gram-positive bacteria, but that has been replaced by D-lysine in gram-negative bacteria. Lipoteichoic acids are being present in the case of gram-positive, but absent in the case of gram-negative bacteria. And the nature of the periplasm and the periplasmic space has been found to be different in the case of gram-positive and the gram-negative bacteria. With this, we come to an end of the cell wall of gram-positive and gram-negative bacteria. Thank you for your cooperation, and if you have any doubts regarding this, you can just put your comments in the comment box so that I can reply to it. Thank you once again.